Hello, welcome to BioFree. As you can see that the slides look a lot different because I'm using my own slides. When I was recording the reproductive system from the last lecture, I found a lot of issues. And when I was trying to record the cell division lecture, I also found a lot of issues. So I decided to use my own slides. It will be simpler and clearer. That's why it took me so much time to record the last lecture. For cell division, aka also known as mitosis and meiosis, I will start using my slides in the last few lectures. And for the word meiosis, some people call it meiosis. Well, so if you hear some people say it meiosis or meiosis, okay, it's the same thing that we are going to talk about now. So how do we grow? As you can see that, well, from a baby to an adult, you know that the body size increase a lot. And also, how do baby or the people grow? How do baby grow to adult? And as you know that when people go to gym to work out, they also get bigger. The muscle get bigger. They get buffer. So how do they do that? What happened? So when we work out to train our muscle, to make our muscle bigger, it is the increase in size of the cell. Our muscle cell become bigger. It is in terms of quality. If some of you do pump iron in the gym, you know that okay, we are supposed to use the weight to tear our muscle cell, so that during the recovery of the muscle cell, this muscle cell will become bigger. So that's why bodybuilder or people who go to gym, the reason they get bigger, they grow bigger because of the size. Another one is increasing the number of the cells. It is in terms of quantity. So that explains why baby can grow as big as adult, because the number of cells inside the body increases. The number of cells increases is using a mitosis, which is the first topic we will talk about, mitosis. And mitosis has another function. We call this asexual reproduction. We talk about that, asexual reproduction. Asexual reproduction, do you still remember what living organism uses asexual reproduction? Well, I'll give you 5 seconds to think about this. 5, 4, 3, 2, 1. Okay, time's up. Yes, asexual reproduction is used by prokaryote, like bacteria. They do it like this. For bacteria and prokaryote, they call this binary fission. And other than prokaryote and bacteria, okay, a lot of coral reef, they also use asexual reproduction. Coral reef. Advantage is that okay, it is very fast. Disadvantage is that there's no genetic variance, meaning that if let's say you have an antibiotic that can kill the bacteria, then the entire colony, all the bacteria, they will be killed by these antibiotics because they have the same gene. So cell cycle. Before we start to talk about the cell division, we need to look at the cell cycle. What a normal cell go through. For the normal cell, they most of the time they spend in something called interphase. Interphase is the stage of the cell that spend most of the time. In the interphase, it has G1. G1 stands for growth. G stands for growth. S phase stands for synthesis. Synthesis. What's the synthesis? It's the DNA replication. DNA replication, we call it synthesis. And then G2, of course, growth. And this is the final preparation for mitosis, cell division. And then after, if the cell go to G2 phase, then the cell will start a mitosis, which is shown in here, mitosis, M. Okay. Mitosis has 5 phases: prophase, metaphase, anaphase, and telophase. Okay, after telophase, we have something called cytokinesis. Okay, cyto means cell, kinesis means splitting, so it is a splitting of the cell at the end. But what about the first 4 phases? The first 4 phases, we call it karyokinesis. Karyo, do you still remember what it means? Or where have you seen this word karyo? Well, if you still remember our lecture, we keep talking about prokaryotic cell and eukaryotic cell. Karyo, it is the same karyo. What does it mean? It means nucleus. So the first step of cell division is the splitting of the nucleus. And then after that, we will have cytokinesis, which is a splitting of the cell. So we split the nucleus first, and then we split the cell. Okay? Now, let's take a look at the details of this slide. You have this G1 checkpoint, G2 checkpoint, M checkpoint. What are those? Well, during the normal growth, during the G1, the growth phase, if there's any issue, such as a mutation occur, then the cell detected, then at the G1 checkpoint, it will stop. You will stop the cell from going from G1 into S. Why? Because during the S phase, the synthesis phase, DNA is gonna replicate. So if there's any issue detected in the growth, G1 growth phase, then it will not go to the DNA's replication, because you don't want to amplify, or you don't want to replicate the mutation, right? And then the G2 phase is at the end of the G2 check. The G2 checkpoint is at the end of the G2 phase. Why? Because if the cell detects any mistake or mutation at the end of G2 phase, then it will stop there. It will not go to the mitosis. Why? Of course, mitosis is the cell division. You don't want to, again, amplify or replicate the mistakes that detects in G2 phase, so that the 2 daughter cell will share the same mistake or mutation. Last but not least, the M checkpoint. There is a checkpoint in the mitosis, so that during mitosis, if there's any mistake in terms of splitting the nucleus or splitting the cell, then you will stop there. Again, you don't want to amplify or replicate the mistake in the 2 daughter cells. Then what about G0 phase? G0 phase is a phase that cell will go from G1 into G0, so that it will never go into the S phase. So the cell will not go into the synthesis, DNA replication. An example of this cell that goes into G0 phase, that means the cell, they don't divide. You don't go through DNA replication or divide. Oops, sorry about that. So an example of the cell that they don't divide is the neuron. Neuron, they are nerve cells, okay? So the cells in your brain, they don't divide, they don't go through the cell cycle. They mostly stay in the G0 phase. Okay, forget about this number 4 here. It's not necessary. So the main point for this slide is that, okay, we call this one chromatid. We call these two sister chromatids. Okay, two sister chromatids. Okay, so when we will see one chromatid? When would you see two chromatids? Okay, so chromosome consists of one chromatid. Okay, this is good. This is chromosome consists of two chromatids. For this one, it is two chromatids, sister chromatids. So when would you see chromosome with one chromatid, and when will you see chromosome with two chromatids? Is that after DNA replication. After DNA replication, then you have two sister chromatids. Okay. Then, when would you go back to one chromatid? After mitosis, after cell division, actually. At the end of the cell, I would say at the end of the cell division. And at the end of the cell division. For example, at the end of mitosis, you only have one chromatid in each chromosome. But after DNA replication, you have two chromatids in each chromosome. Okay, I want you to be able to tell the differences between chromosome and chromatids, okay? And then last but not least, I want you to learn this term centromere. Centromere is the center part of the chromosome. It is the part that connects the two chromatids, okay? During the cell division, when we are talking about mitosis, we will talk about something called a centrosome. And centrioles. So if you still remember the beginning of semester, we talked about cell structure and function, the centriole and centrosome. Then you know that those are the one of those cell organs, so cell organelles. Okay, I will review it for you when we go to the mitosis. But keep in mind that we have three very similar terms: centriole, centrosome, and centromere. But right now, just memorize centromere. Centromere, they are the connection between the two chromatids. They are in the center of the chromosome. Okay?